Welcome to NeuroSociety Stories, a series created by the Dana Foundation that explores the incredible ways that neuroscience is shaping society. Through engaging conversations, we invite Dana Foundation grantees, partners, and other neuroscience and society thought leaders to share stories of discovery, innovation, and impact. Each episode offers unique perspectives on how brain science intersects with fields such as ethics, law, social sciences, and more, advancing neuroscience for the good of all. I'm Caroline Montojo, President and CEO of the Dana Foundation. Join us as we uncover how neuroscience can drive change, inspire new possibilities, and create a future that reflects the aspirations of all people. Hey, Serge.
Hey, good to see you.
Good to see you too. Thank you so much for coming in.
Thank you. It's great to be here.
Please have a seat.
Thank you.
Take one, Mark. Today, I am delighted to welcome world-renowned neuroscientist, Dr. Sergiu Pasca. He was trained as a physician in Romania, and he's now a professor of psychiatry and behavior at Stanford University. He's also the director of the Stanford Brain Organogenesis Center. Sergiu and his lab have played a key role in the development and use of neural organoids, 3D clusters of brain-like cells grown from human stem cells. Sergiu and his team are pioneering new uses of neural organoids and assembloids to better understand human brain development, psychiatric conditions, and potential therapeutics. We're also delighted to talk with Sergiu today about his interest in the ethical and societal questions posed by this area of research, as well as new strategies for charting a future of responsible innovation and science communication on neural organoids. Sergiu, welcome to NeuroSociety Stories.
Thank you so much. It's great to be here.
You've said that your interest in neural organoids began as a way to understand the brain outside of the human body. Can you take us back to that moment? What drew you in, and did you ever imagine that the field would grow as rapidly as it has?
Initially, I was just puzzled for a while, and my approach really was to try to measure anything biological in this condition. So I studied what changes are taking place in the blood of patients with autism. Autism remains sort of my main clinical expertise. And then suddenly, as I was finishing my clinical training, I've heard of this remarkable discovery that a Japanese scientist made, showing that you could actually take skin cells or any somatic cells, turn them back in time to become stem cells, pluripotent stem cells. And of course, pluripotent stem cells can become any other cell types. So then the idea was immediately, could we actually, in a non-invasive way, without taking a biopsy, without doing anything to patients, just turn skin cells into neurons and study the biology of those psychiatric disorders outside of the human body? And so I really dropped even the clinical training that I was doing at the time and moved to the United States and came to Stanford, where I developed soon after, in the next few years, some of the first neurons that were derived from patients with neurodevelopmental disorders. And that really unleashed so many possibilities. And of course, over the past 15 years, we've been building ever more complex models of the human brain outside of the human body with the clear intent of understanding the biology of the psychiatric disorders and really demystifying psychiatric disorders so that we can ultimately find effective therapeutics.
That's incredible. It's wonderful to hear that story of how you first discovered this area and then translated it into neuroscience. You were thinking about ethics long before neural organoids started to get combined into assembloids and before they were transplanted into animals. How did your interest in ethics and philosophy shape the way that you approach your work?
I think all neuroscientists are interested at some level in philosophy. We're interested at least in the philosophy of mind, like how does the mind arise out of the brain. So it became very clear, especially for us in psychiatry, that understanding the biological basis of how mental processes really arise out of circuits, it remains a fundamental question. And then of course, I came back to that as we were building some of this model outside of the human brain. And some of the questions arose naturally. And then there was also a much more pragmatical aspect of it. we're using human cells that we're obtaining from patients with the consent. And so we have to be responsible in the use of those cells. And so, of course, the convergence of these two issues, I think, sensitize us very early on about these issues that are, I guess, more prominent at this point.
Thank you. We're now at a point where you can watch neurons develop in a dish. Can you share what kinds of possibilities this opens up, and what are some of the biggest ethical questions that arise from these breakthroughs?
In the beginning, most of the cultures that we're deriving from these pluripotent stem cells were rather simplistic, bidimensional, like sitting at the bottom of a plate. We realized very quickly that in order to understand some of these complex psychiatric disorders, we would need to gain access to later stages of development where the brain starts to essentially assemble itself and form the circuits that are so critical for brain function. And so we essentially embarked on this journey. And initially, we started trying to see how many cell types can we actually make in the brain. Of course, there are more than 2,000. But now we're in a very good position where we can generate many of the cell types in the nervous system. And then it turns out that most cells in the nervous system don't stay in the place in which they're born, but very often move very, very long distances. And so then we started to create preparations, and that was the first actually assembloid, where you make two brain regions where some of the cells have to move onto the other one. We put them in close proximity, and it turns out that the cells know what to do. And then, of course, in an attempt to try to model some of the circuitry, we also started to put more than two parts together to really see to what level the cells connect with each other. And again, what we started to discover, which is really remarkable, is that to a large extent, the cells know what to do. They come with instructions, and then they start to form on their own some of this circuit. And so, for instance, you can rebuild the entire cortical spinal circuit by putting a cortex, a spinal cord, and a ball of human muscle. And then the ball of human muscle will start to contract in a dish once you put them together. And I think this speaks to how remarkable the assembly of the human brain is. Now, about the ethical issues, it became also very clear to many of us in the neuroscience community over the past few decades that human brain disorders or human psychiatric disorders, they have a very strong human component. So it's clear that we need to get access to the human brain and recreate some of the circuits. But of course, the closer we get to it, the more ethical issues arise. And most of them are arising because we now know that accessing the human brain will likely be key to understanding some of these conditions. And yet, the closer we get to recreating some of this process, or the more human these preparations are, the more uncomfortable we also feel about some of these issues. On one hand, there are the issues of the source of the cells. The cells are obtained from individuals under consent. And so there are certain limitations of what can be done with those cells. And of course, there are the issues of how complex some of these preparations are becoming. If an assembloid becomes really complex, could it actually have new emergent properties? Could it actually have sentience? And that is one of the issues that is being discussed quite broadly right now. And then, of course, there are also the issues related to transplanting the cells themselves into animals and thinking about the welfare of those animals. But again, all of these issues, I think, have to really be put in the context of why we're doing this type of work. Psychiatric disorders bring immense suffering. 1 in 4 individuals suffers from a brain disorder worldwide. And yet we're not making much progress in finding effective therapeutics. So I think we have to think about what the goal is for most of this technology and how we're going to actually deploy it.
You have called for a shared ethical framework around this new frontier of neuroscience, one that balances both responsibility and innovation. Can you share your thoughts on what a healthy field culture would look like?
Well, we realized very early on that even what we call these preparations, it's important. If we trivialize what we've made and let's say we call them mini brains, that will only confuse the public. And so the first step was actually to get together with a larger group of experts around the world and really convene on a set of terms that would accurately describe what is being done. So that was the first step. The second step was actually for us to get even a larger consortium and then discuss what are some of the standards for the field. So that when we talk about experiments, we're all talking about the same type of experiments. And that was step number 2. Now that we've clarified the terms and we know what some of the experimental conditions are, then we should start thinking, what are the real ethical issues that are arising? And this is really, they're arising as these experiments are done in various laboratories around the world. And I mean, just our center at Stanford and my lab has helped more than 350 labs around the world implementing these technologies. The need is immense because you can, for the first time, ask questions not just about how the human brain develops or what makes it different than other brains, but really to ask very specific questions about how disorders are arising and how to develop therapeutics. So this is really a prime time for getting together not just the experts in the field, but philosophers, ethicists, legal scholars, journalists, science communicators, parents of these patients who really have a stake in like how this technology is really being used, and discuss openly what that framework should really be moving forward. And that's why it's so exciting to partner with the Dana Foundation in this effort later this year.
That sounds like a very healthy field culture that you describe, with all of these different people coming around the table to talk with each other about what the future of neuroscience can look like. I'd love to hear more about what this future Asilomar conference you're organizing will look like, and how do you hope that it will shape the field?
Our goal was really to bring everybody together first and really have a healthy conversation about the real issues are. So our hope is that we will present an overview of the science as it stands today or as it stands in the fall. So we'll bring a series of experts in neuroorganoids and assembloids and their transplantation. And then we're going to have philosophers, journalists, legal scholars, sociologists of religion. Our hope is to really bring a very broad perspective and really identify the key issues that one should be aware. And at the same time, the next step will be to really engage some of the scientific organizations, such as ISSCR, Society for Neuroscience, in helping us really put together a broader framework for the entire field.
And I think you also had mentioned the inclusion of patient groups. Can you give us a sense of the different patients and families that you're bringing into these discussions?
Yeah, absolutely. I mean, from early on, we noticed that parents, especially as we work with neurodevelopmental disorders such as autism and intellectual disability, the parents are very engaged. And of course, parents would love for us to develop therapeutics with this technology, but at the same time, they're just aware of how this will probably transform the field for the next decade. So I think we have to just think, what are really all the serious issues that may be arising at the ethical level, and then create the right framework that is needed for the field to progress in a healthy way. But yeah, parents of patients are actually key to this conversation, in my opinion.
So you've shared about how far this field has come over such a short period of time. Can you share your outlook on where it's going and its potential in therapeutics, helping people?
It's so important to think about what ultimately the potential of the field is. And I think the example of this form of autism called Timothy syndrome that we've now been studying for 15 years. This is a rare form of autism and epilepsy. Very, very rare. There are just about 20, 30 patients worldwide. And yet, this disorder is giving us key insights into how we could develop a therapeutic using this platform. And over the past few years, we've actually developed a first therapeutic for this condition that we hope to move into clinical trials sometimes early next year. And this will be the first therapeutic for a psychiatric disease that has been exclusively developed with human stem cell models. Of course, it may fail, and this is just the first disease, and we have about a dozen of these conditions that we've been studying from various angles. But we will not stop at this point. This is really the best of times to study human neuroscience. And I think it's the best of times to really understanding the biology of these complex disorders and really find therapeutics.
It's wonderful to hear about your outlook for potential therapeutics using neural organoids as a model. That's exactly what we are interested in here at the Dana Foundation under our Neuroscience and Society mission. We hope to be able to seed new projects, new programs, and people who are interested in education, training, and bidirectional public engagement on neuroscience and society issues like neural organoids or brain-computer interfaces. So it's wonderful to see that connection of neuroscience being studied in the lab and then thinking about that bridge into how it can positively impact people's lives. So thank you so much for the work that you do.
Well, thank you. I look at it more of a convergence, if anything, because I think really partnering with the expertise at the Dana Foundation it's key, I think, at this point as we move forward in this field.
Sergiu, this has been an incredibly fascinating conversation. I've heard you talk about your research with neural organoids, assembloids, and its potential to really help those with psychiatric conditions in the past. Every single time I talk to you, it feels like the field has leaped forward multiple steps. And so I want to thank you for being so intentional and thoughtful about the ethical and societal questions that arise from this work. I'm also looking forward to the Asilomar conference that you're organizing and all of the outcomes that come from that, because it's very unique how you're bringing together people from so many different fields, as well as patients and their families, to chart that path forward. So thank you for your work.
Yeah, thank you so much. Thank you.